Hi everyone, welcome to the final live Q&A of the day. Today I've got with me Shane Hegarty and Graham Dempsey. Thank you for joining me, both of you. My first question is for Graham. What is the biggest differentiator of Bright over existing platforms when it comes to the discovery of therapeutic candidates for CNS?
Yeah, that's a great question. I think where we have focused has really been around integration of human cell-based models, really measurements of neuronal properties and biology, at the scale of many billions of neurons, and then the computational tools that allow us to really understand disease phenotypes and pharmacology in a very quantitative way. So I think that that's important. Part of our platform has been focused on optogenetics, and this allows us to use light-based methods to measure the function of neurons with about 30,000-fold higher throughput than other approaches, like manual patch clamp electrophysiology, or even multi-electrode arrays and calcium imaging. So I think the scale and throughput and then the integration of those components is really important.
Okay, thank you. And then Shane, how could you apply your AAV CRISPR screening platform to other neurological disorders?
It's a great question, and it really depends on the biological question you're interested in and the phenotypes you can study with available animal models. And once you've decided on the animal model, then the question is there an AAV serotype and delivery method optimized for you to be able to target that tissue of interest? I gave an example in the presentation, potentially an ALS mouse model where you could use your AAVs, maybe AAV9 or AAV-PHBB, to target the spinal cord and central nervous system with a reasonable efficiency that you would get your CRISPR delivery to your target tissues. And, at the time point of interest as well, the reason we go through the trouble of using these AAVs is because you can, deliver them to the adult mouse or, at a time point relevant to the disease, and then you avoid caveats of, having a knockout mouse where there could be a lot of developmental or abnormalities that interfere, and confound your results later on when you go to do your disease modeling. But of course, we don't have an AAV serotype for every cell of interest. There's a lot of advances over there. You can use cell type specific promoters if you like. The more complicated you make it, I'm sure the more difficult it's going to be in practice. That's always a consideration, especially when we think about scalability, which is a massive limitation for in vivo screening. It's really just how many pairs of hands do you have, or how good you're at the model. It's not as sophisticated as Graham's approach, for example, but there's a value in it that sometimes makes the effort worth it, depending on what you're interested in trying to treat.
Indeed, thank you. And we've had a question put in the chat. Does the lack or limited disease phenotypes observed in iPSC-derived neurons modeling CNS disorders? It's time to look for new models for target validation. And then they've got a follow-up question, which in the future, how useful do you think iPSC-derived neuronal models will be?
Yeah, I can start there. I think that any technology as it evolves, I think people start to understand its utility and where the places are that it's ready for prime time in terms of application. So I think in our experience there have been many phenotypes that we have observed. I talked about this a bit in my talk, giving examples from genetic epilepsies or monogenic disorders broadly, where you can use CRISPR gene editing. You can introduce mutations very precisely. You can do really nice comparisons of well-matched controls with disease states. And our experience has been that we have really in all cases have identified phenotypes. And that's just because, as you'd expect, disorders of the brain almost certainly manifest themselves through the function of neurons. So that's been our experience. I think there's a lot of emerging literature where people are having success in terms of applying those tools. And I think absolutely the applications of the technology going forward are really exciting. In the context of being able to create, these are what I presented on are two-dimensional representations There's a lot of work going on in terms of pressing these into more complex co-cultures of human cell types. So different cell types, different neurons, different supporter cells, and also a lot of work going on in creating organoids or assembloids that are really trying to continue to get at the three-dimensional nature of the brain. So it's our view, and I think shared by many, that having a human cell-based model of the brain and disorders is going to be a really important component of therapeutic discovery. And I think there's a lot of exciting future. Anyway, that's my point of view.
Shane, would you like to add anything at all?
Sure. It's hard to follow Graham. He is the expert in the area, and I agree with almost everything he said there. And obviously, if Beth was here, she'd have a great perspective and overview as well about what we can achieve today with our current tools and differentiation protocols, and how they're trying to address 3D nature of the model, or multi-systems interacting in some device like microfluidics, for example, potentially. And I would say without much experience in the space, the more complicated the model is, maybe it makes it even more difficult to study your phenotype. We always have to know, what are we actually phenotyping here, and if it is disease relevant. And that's really the most important thing. Obviously, from there's more translational work to be done. So what are you using those cells for? At least you get to study human biology, human mutations. That's huge, right? That's why they're so powerful. And then once you know what you're measuring, and you can do it reproducibly, the limitations of it, then the next steps later. After you've validated and you've identified your hits, you can take the next step. So you don't have to do everything in one model. That's naive. Nobody here is suggesting you should do all your work in one model system. But it's amazing the progress that's been made, and I'm very excited to see where it goes.
Amazing. Hi, Beth.
Hi.
Beth. Hi, thank you for joining. We just had a question that was put in the chat box, which I'll repeat for you. Does the lack or limited disease phenotypes observed in iPSC-derived neurons modeling CNS disorders? Is it time to look for new models for target validation? And then they also had a follow-up question of, in the future, how useful do you think iPSC-derived neuronal models will be?
Okay, let me start with, I don't believe that there aren't phenotypes. I think you have to choose the right cells. So in other words, in my case, in HD, I think a lot of people have chosen the glutamatergics because they're easy to make. The medium spiny neurons are much harder to make, but they are the right cell. First, we already know that those are the most sensitive. They die first. This is not to say the cortical neurons, the glutamatergics, are not important, but you have to go where the phenotype is. You have to follow the disease, not what's easy. And so in medium spiny neurons, we actually see quite a lot of phenotypes. The other piece I'd say is that the glutamatergics do show phenotypes. For instance, if you go out further to 35 to 38 days in culture, you can actually see some very beautiful electrophysiological differences using the multi-electrode array technique. So I think it's a matter of, that would be great, and we would look at that, but only once we had a handful of compounds as opposed to a bucket of compounds. And this is really what I mean by choosing the right model with the right complexity for the question you're trying to ask. So that's what I would say about that. Do I think iPSCs are going to continue to be useful? I do, unless we find another way to make neurons. I'm very interested in the fibroblast, in the direct differentiation from fibroblasts to neurons, but the challenge is you just can't get enough of them to really use them at an early stage where you're trying to screen potentially through thousands of compounds. And so it's always an issue of, it's like almost anything. It's an issue of, it's not only this or only that. It's how do you put everything together to do the job, the question you answer, the question you need to answer right here, and then you bring in the next models. And a lot of that, as I showed, has to do with throughput. So I hope I've covered everything that was asked in that complex question.
Yes, thank you very much. And then back to you, Shane, a question for you. What other disorders could be treated with your KCC2 drug?
It's a big question. We initially focused on spinal cord injury and its associated comorbidities like neuropathic pain and spasticity because our expertise and the data was driving us in that direction. We're very excited about that indication, but we're not kind of ignorant of the other potential indications or patient groups that might benefit from such a treatment. There's a lot of, for example, treatment-resistant seizures where you might re-enable endogenous GABA to restore some inhibition to those circuits or even enable the activity of other drugs as an adjunct. So we're very interested in that direction, and we've started to, in collaboration with a number of organizations, to try to build this preclinical data set in the relevant indications for KC2 using our first-in-class oral KC2 potentiator drugs. And, we're very lucky. I know that's probably, again, not up to us. We did our screen in spinal cord injury, weren't really thinking about KC2 at all. It just happened to be one of the compounds we could use. And then we've discovered KC2 and its biology. And there's a whole field there, about a decade of work that keeps growing in many different directions. And we're lucky to be able to build on these different experts discoveries on that target and its role in different disorders, and try to see where our pharmacological approach, is it relevant for that particular pathology in that patient group. Not every KC2 pathology is identical, so we spend a lot of time trying to figure out with our approach, the pharmacological approach, what is that useful for and at what time frame and in what setting. So we're very excited about that. We're a biotech company. We can't do everything at once, but we try to do a lot of indication prioritization exercises and scientific exercises, and collaborating and speaking with KOLs to really know what's the best next step now that we're looking at these other indications. But yeah, we're very excited about the potential there, and even for AAV-KC2 as well as another modality for restoring KC2 hyperfunction in certain settings. But there's a lot of interest there, and I'm excited to in the future be able to show people what else we can do with these therapeutics.
Amazing, thank you. And then Graham, could you outline for us the next step, your future applications for the Bright5 system?
Yeah, absolutely. As I mentioned in my presentation, there's been a lot of work really focused on, monogenic disorders in terms of seizure and neurodevelopmental disorders. And we're continuing to progress on that front, actually leveraging the Bright system for our therapeutic discovery applications. That being said, I think there is a lot of exciting technology development that we're pursuing, as well as applications to new areas like neurodegeneration. What we really try to focus on, which I think Beth said very nicely, is what is the disease relevant cell type, and then really being able to generate data sets in the disease relevant cell type where we're looking at the functional consequences of perturbing various genes in the genome, and how that can then allow us to actually map phenotypes and pharmacology. So I talked a little bit about this in the presentation, thinking about the ability to create what we call drug fingerprints, which would allow us to do really in silico prediction of matching compounds or matching targets to specific phenotypes. So we continue to progress on that front, and also really progressing on continuing to integrate the complexity of other cell types to really start to open up new indications, areas in neurodegeneration, neuroinflammation, which I think are really exciting potentials for us. And then, in the context of different therapeutic modalities, we are doing a lot to use our platform for small molecule and antisense oligonucleotide-based discovery. But we think that there are exciting applications to using our approach in the context of things like gene therapy and gene editing, where one really wants to start to understand at a single cell and cell-based level the connection between phenotype and specific molecular readouts like protein expression, things like that. So I think that's kind of where we're focused right now.
Mm.
And yeah, lots of exciting future potential from our perspective.
Amazing. I've got one final question just because we're close to time. Beth, would you mind telling us how complex are your disease models and how many cell types does a disease model need to include?
I think if I knew the answer to that, I'd be really rich. But seriously, our initial models that we use are really just single cell type for simplicity. What we've done with the spheroids is to, these are cortical spheroids, and we're really just starting to analyze those, is combine both glutamatergic and GABAergic neurons with astrocytes, and these are all human. And the reason we decided to go after the spheroids rather than the organoids is really an issue of reproducibility, and because you can make this pretty high throughput. So I think the organoids are fantastic. I think they're not quite as high throughput, and I'm not sure how incredibly reproducible they are. I know people are really interested in working on them. I think they're very interesting as well. I know that also there are people, I showed sort of an organ, a body on a chip kind of thing. And I know people are working on that. But, that's the way I look at it in terms of drug discovery is when I have, one or two compounds I think are really interesting, I might collaborate with someone to work on that. I don't see, a small biotech like Origami Therapeutics work, generating those sorts of models. And I think you can see from my collaborations that I've tried to, basically leverage what other people have done, and that's where I'm really grateful to some of my collaborators like Scintillant, because they're willing to take on something that's a little bit researchy and work through the challenges. So again, I think it depends what you're trying to ask. If you're asking the sort of thing about circuits, then you need to set up those circuits. And I think there's some really nice hardware, if you will, for really setting up some of those in a very defined way, where you can grow neurons and have them, synapse on each other and look at that circuitry. So I guess I'll stop there and just say, again, it really depends on what question you're trying to ask. And obviously you want to get more and more complex, but if you start there, you need a big fat computer and a couple of very expensive data scientists to sort it all out. And I would say really important, I would love to have a data scientist, but to start with, you can start in a simpler model.
Amazing. Thank you very much, all three of you, for joining me today. We'll have to end the Q&A there, I'm afraid, as we've reached time, but thank you very much for joining.
Thank you.
Thank
you.
Thanks.
Bye.